Students, welcome to Biotech Academy. Today we'll study about peptide bonds, the characteristics of these bonds, and about Ramachandran plot, which is used to determine the type of structure which is formed by the amino acids. Alright, so as discussed in my previous lecture, that amino acids are the building blocks of proteins which are connected to form, which are linked together to form a polypeptide via specific type of covalent bonds that is known as peptide bond. So I've used two terms over here. One is polypeptide and the other is peptide bonds. So first we'll define these two terms. So what is a polypeptide? A polypeptide is a linear unbranched polymer of amino acids which are connected via peptide bonds, which are a specific type of covalent bonds. And what are peptide bonds? Peptide bonds are the amide linkages which are formed when the unshared electron pair of alpha nitrogen atom of one amino acid. Okay, so alpha nitrogen atom of one amino acid which has an unshared pair of electron attacks the carbonyl carbon atom of the other amino acid. So it is taking two amino acids into consideration from one nitrogen amino group nitrogen atom and the other carbonyl group carbon atom. And when there is amide substitution reaction, then this covalent bond is formed, which is known as peptide bond. And in this bond, one molecule of per peptide bond formed between two amino acids, one molecule of water is released. Also, the delta G, which is the free energy change of the reaction of the bond formation, is positive, means it is an endothermic reaction, means it is utilizing energy during the bond formation, it is consuming energy. So let's discuss in detail about all these things. So first, how the peptide bond is formed. First, if you remember, the general structure of an amino acid, which is like, it has a central alpha carbon atom, it has a carbonyl group, it has an amino group, it has an R group which is variable in all 20 standard amino acids, and a hydrogen atom. So this is the general structure of an amino acid, and there is an unshared electron pair over here in the alpha nitrogen atom. This unshared electron pair attacks the carbonyl group of the other amino acids So one amino acid nitrogen is involved, the other amino acid carbonyl carbon atom is involved, and one molecule of water is released. So how? This is the general structure of an amino acid. So let's now let us discuss about how the bond is formed. So first, if you last time I told that all the 20 amino acids have different R groups. All the 20 amino acids, in all the 20 amino acids, the R group is variable. Rest of the structure remains the same, while the R group is variable. So suppose there are two amino acids, amino acid 1 which has an R group 1 and amino acid 2 which has an R group R2. So how the bond is formed between these two, let's have a look at that. First we'll draw the amino acid structure. Amino acid first, C alpha N H R1 H C double bond O H. Okay, so this is amino acid 1. Alright, and the other amino acid will be with central carbon atom alpha, H, R2, N, H, H. So I'll ask you to consider these important things. Why have I written these R groups in opposite orientation in the two neighboring amino acids which are going to form a peptide bond? That you keep this thing in mind. I'll describe why the structure, why the positions are like this in few minutes from now. So one molecule of water, there is a electrophilic amide substitution reaction, and one molecule of water is released, alright, and a bond is formed between N and C. This water molecule is released, so the peptide bond formed is and this bond, this is known as peptide bond. Alright. Now coming to delta G, which is the free energy change. So you must have studied in your 11th-12th standard. Delta G is the free energy change. Free energy change, the value of the delta G, it determines what type of reaction it is. Whether it is an exothermic reaction or an endothermic reaction, or the reaction is at equilibrium. So it can have three values. Okay, it can have positive value, the delta G can have 0 value, or it can have negative value. So if the delta G, if the value of delta G, that is the free energy change, when reactants combine together to form products, when that the free energy change, if that value is negative, means it is an exothermic reaction, that is energy is released in the process. Whereas if the delta G value is positive, means it is an endothermic reaction. Endothermic reaction is one where energy is consumed to form the product. So the reactants consume energy to form the product, whereas if the delta G value is 0, means the reaction is neither occurring in the forward direction or in the reverse direction. So the reaction is in the state of equilibrium. So that is all about delta G. Okay, so for the peptide bond, it is a condensation reaction, plus it is an endothermic reaction where the delta G value is positive, means it is consuming energy during this kind of bond formation. Okay, now I'll erase this part. However, I'll keep this structure like this. So this is the peptide bond. Okay, now this NC bond, this NC bond is very important, and it imparts the specific characteristics of the peptide bonds because of the property of resonance. Means it has partial double bond character. So the peptide bond has partial double bond character. So this is like this. And why this? Because of the resonating electron, means it has partial negative, this is partial positive, and it is not static. The electrons are not static over here. Okay, so because of this partial double bond character, the bond length of this NC bond is shorter, and this bond length is 1.33 Armstrong. Okay, because of this partial double bond. Whereas usual NC bond, if the bond is single, if there is single bond between nitrogen and carbon, the usual bond length is 1.45 Armstrong. Whereas because of this partial double bond character in the peptide bond, this bond length is reduced to 1.33 Armstrong. So you remember. So there are few things which you must recollect from your 11th and 12th chemistry. So as the number of bonds increases between the two atoms, the bond length decreases. So single bond large, double bond little lesser, the bond length is slightly less, whereas triple bond the bond length is shortest among all the three kinds of bonds. Okay, so because of this partial double bond character of the between nitrogen and carbon, the bond length is reduced to 1.33 Armstrong, whereas the usual single bond length of between nitrogen and carbon is 1.45 Armstrong. So this is one of the important property. Now again, because of this partial double bond character also, why this partial double bond character, which is ultimately because of the resonance, it holds, there are six atoms in the play of the peptide bond. So 1, 2, 3, 4, 5 and 6 means 2 alpha carbon atoms, 1 NC, 1 oxygen and 1 hydrogen. So these are the 6 major atoms which are forming the peptide bond, and this partial double bond character of between nitrogen and carbon, it keeps all the 6 atoms in one plane. Alright, so it is planar. So the peptide bond is planar in nature. Okay, so because of this partial double bond character. Now the third property of this is that when the two neighboring amino acids which are forming the peptide bond, because of this double bond character, the rotations here are not allowed. However, there is rotation allowed between NC alpha and C alpha bond. Okay, in theory, for all practical, it can have all possible values because they are lying in one plane, so it can have all possible values between minus 180 degrees Celsius to between minus 180 degree to plus 180 degree. Alright, we are talking about angles, not temperature. So minus 180 degrees to plus 180 degrees. So it can have any value between minus 180 degree to plus 180 degree. The bond rotation between NC alpha and CC alpha. So theoretically, this rotation is known as phi, and this angles are known as psi. These are termed as phi angles, and this rotation's values are known as psi angles. However, all rotations are practically not possible. Now, why all rotations are not practically possible? Because of this steric hindrance. And what is steric hindrance? Steric hindrance is when two atoms come in such close vicinity to each other that they are lesser than their van der Waals forces, van der Waal interactions or van der Waal radius, such that you can say there is increase in the randomness or disturbance, and it is sterically not allowed. You can also say that two lions cannot exist in one forest. So it follows the same rule. So all values are not allowed. Theoretically, it can have all possible values. However, there are certain restrictions between different angles because of these steric hindrances, and steric hindrance depends on these or the side chains of the corresponding or the neighboring amino acids. Now these amino acids, these are always in trans. The two amino acids which are forming, the neighboring two amino acids which are forming a peptide bond, there the R group always occupies the trans position. So if you remember chemistry again, class 11th and 12th, there are two forms, cis form and transform. If the two, suppose this is the bond, and if this is the bond, if the two positions or the two groups are lying in the same plane or above, so that is cis configuration. Whereas if they are lying in the opposite directions or opposite planes, one up and one downstairs, so that is trans configuration. And so in case of neighboring amino acids, the R group always occupies the trans position. And why trans position? Because cis position, again, if for the larger atoms, because R group varies in different amino acids. So for larger amino acids, the number of carbon atoms are more, so they come closer, lesser than the van der Waals radii, and it creates steric hindrance. So the amino acids, the R groups of the neighboring amino acids always occupies the trans form. Okay, so now we'll discuss about what are the permitted values of phi and psi angles. And first time the permitted values of phi and psi angles were described by Ramachandran, and therefore it is known as Ramachandran Plot, where all the permitted values of phi and psi angles of the amino acids and proteins are plotted on a two-dimensional map, and it is known as Ramachandran Plot. So we'll discuss about now we'll discuss about Ramachandran Plot, which describes the phi and the allowed values of phi, which describes the allowed values of phi and psi for various amino acids and proteins. Alright, so this is, I am drawing here the Ramachandran plot. Okay, so it is a two-dimensional plot with phi and psi angles. So it is minus 180 degree here, 0 plus 180 degree, then again 0 plus 180 degree. This is psi and this is phi. Okay, now this is beta sheet. So all values lying here in the shaded regions are beta sheet. Then there are certain areas over here, then this is right-handed alpha helix, and here it is left-handed alpha helix. Alright, so now I'll describe this, explain what is this all about. This is the Ramachandran plot where the phi and psi values are plotted on two-dimensional map from minus 180 degree to plus 180 degree, phi, and psi again minus 180 degree to plus 180 degree. Now over here, whatever the regions, these are some of the marked regions. Apart from these regions, in the rest of the regions, these corresponds to conformations where the phi and psi in these phi and psi angles on in the polypeptide chain, when forms part of the polypeptide chain, the atoms are in too close vicinity and they're in so close proximity to each other that they come even lesser than the van der Waals radii and cause steric hindrance. So these apart from these marked regions, the rest of the regions are practically or sterically they are not allowed, or conformations are not allowed. The shaded regions, now these the rest of the regions that sterically not possible or conformationally they are not allowed, because except there are two exceptions for the rest of the regions. These two exceptions are two amino acids, one is glycine and the other is proline. Now why glycine and proline? Because in glycine the R group is single hydrogen atom, so and also the size of that atom is too small, so it doesn't cause any such hindrance between the polypeptide backbone or between the neighboring atoms. In case of proline, because it has a 5-carbon ring, so the 5-carbon ring the R group has a cyclic structure. Again, here also the steric hindrances are very minimal between the polypeptide chains backbone or the neighboring atoms. So these are the two exceptions for the rest of the regions. However, now coming to the shaded regions, now these shaded regions, these correspond to conformations where there are no steric clashes. Alright, so there are no steric clashes, and these are allowed regions for alpha helix and beta sheet. This is for beta sheet and this is for alpha helix. So that in areas where the density points are maximum for there are two values of phi and psi where the density points for most of the amino acids or most of the proteins, they are high for alpha helix. So the probability of forming alpha helix in those regions where those amino acids are more, those angles are for, if the phi angle is minus 60 degree and psi angle is also minus 60 degree, it generally forms, so minus 60, it has maximum density of forming an alpha helix. Alright, so this is phi is equal to minus 60, psi is equal to minus 60. At these two angles, there are minimal steric clashes among the atoms. Then for beta sheet, the phi angle is minus 90 degrees Celsius and psi angle is plus 120 degrees Celsius. Here it forms beta sheet. So the maximum probability of forming beta sheet is at these angles, whereas the maximum probability of forming alpha helix is these angles. So if you know the values of these angles, you can for those proteins where the structure is unknown, whether it is forming an alpha helix or a beta sheet, these are the secondary structures which we'll discuss in the upcoming lectures. These are the secondary structures. So if you know the phi and psi angles, you can postulate what secondary structure that particular protein might be forming. If you have, so these are used for prediction of the secondary structures of that particular protein for which the structures are not known. So there are certain bioinformatic tools which determine these angles. Alright, now usually glycine and proline are not preferred over here. I have described earlier as well, because the R group in glycine is single hydrogen atom and proline because of its cyclic 5-carbon structure. Next, the amino acids with larger side chains. So the larger the amino acid, the side chains of the amino acids will be, there will be greater chances of steric hindrances, and so the there will be minimal density. So there will be very lesser values of these phi and psi angles where they will be stable. So larger the side chains of the amino acids, lesser dense are the points for forming either the alpha helix or the beta sheet. And smaller the side chains, more denser the points are for forming alpha or beta, alpha helix or beta sheet. Now mostly there are right-handed alpha helix. However, so I have described about the shaded regions. Now what about this other region? This between, like these crosses, these boxes. What about these? Now these are the regions where if we reduce the van der Waals radii, then there may be chances of formation of these secondary structures. So these are for prediction purposes. The last important point about the left-handed alpha helix, where mostly they form right-handed alpha helix. However, because in proteins, remember I told in the previous lecture about L-alpha amino acids. So proteins, they mostly contain, although both L and D and L isomers of the amino acids exist, but in proteins only L isomers are present. So L-alpha amino acids, they mostly form right-handed alpha helix. However, they cannot, these cannot form the L amino acids, the L form of the amino acids, they cannot form extended regions of left-handed alpha helix. However, they can form with, at certain points, there are very less points, but at certain points they may form left-handed alpha helix. So the region is very less for left-handed alpha helix, and it mostly where both phi and psi angles are positive for the left-handed alpha helix. And this is this structure is not very common. Alright, so it was all about the peptide bonds, the characteristics of the bonds, and about Ramachandran plot. So there are certain things, key points which you must remember is about the characteristics of the bond. So how what is the peptide bond? There is a resonance. Because of the resonance, the NC bond is partial double bond character. It has partial double bond nature. Then it is planar. It keeps all the 6 atoms of the peptide bonds in a single plane. Then the R groups of the corresponding amino acids, they exist in trans configuration. Alright, and the bond length between of NC is lesser because of the partial double bond character, and it is coplanar in nature. So these are some of the characteristics of the peptide bonds. Again, it is the formation of the peptide bond is a condensation reaction when water molecule is released. Then you must remember about these two values of where if phi and psi, what are phi and psi angles, and where this plot, if it is given this plot, if you have been given this plot where right-handed alpha helix will lie, where beta sheet will lie, and where left-handed alpha helix will lie. So if you know, also you must know if the phi and psi angles are mostly -60 and -60 both, it has maximum probability of forming alpha helix. However, if the phi angle is -90 and psi angle is plus 120 degrees plus 120 degrees, it has the maximum probability of forming beta sheet. So these are certain key points which you must remember from this lecture, and from this you will get definitely one or two questions are asked in the exam. And for details you must refer to the book, and if you all have any queries, you can ask me in the comment section. I'll be happy to answer all your queries and doubts. So all the best for your preparation, and we'll discuss some of the problems in the next questions which have been asked. Whatever I have taught till now, we'll discuss problems which have been asked in the previous year's exams. We'll discuss in the upcoming video for your practice purposes. So thank you.